This slide is one of those designed to illustrate the nanoscale. This shows a 1 micron scale marker in a human hair. Actually, I measured my own hair, which is only 50 microns in diameter, compared to a neuron in the human brain. I'll say a little bit more about, and comparing it to the size of a bacterium. I'm going to scale that up now by a factor of 1,000 times finer. And now we're at dimensions where this is 10 nanometers. This is what the DNA molecule looks like. This is actually what you might call the basic processor in biology, where a protein sticks through the cell membrane and can perform a function like a switch of either allowing or disallowing an ion to go through. So in biology, the switch is really small at the nanoscale. This, of course, is the carbon nanotube and the experiments that were talked about before, moving individual atoms around. I like to point out that we have living proof of the importance of nanostructures because everything of importance that goes on in a living system goes on at the nanoscale, both the information and the important processes. And to think about or to speculate on what's going to happen in nanotechnology in the future, we can look at, I think, at what living systems do, because these are the ultimate nanoscale-based systems. So let's look at the function. If we look at the functions of computation, memory, material synthesis, and so forth, in some cases we can do much better in artificial systems. Computers go much faster, have much better memory than any human being, and so forth. However, I can go meet somebody on the street that I went to school with when I was 8 years old, and even though they're turned gray and they may be a lot heavier, I can recognize that person because the way in which my brain works in a very different way than what a computer works. And these are some of the points you were making. There are areas where artificial systems do much, much better. For example, electromagnetic detection, absolutely better than a living system does. But there are other things which living systems do extremely well, and I raised this point a few minutes ago, namely catalysis, enabling reactions, chemical reactions to go forward that do not want naturally to go. And we do this much, much better in living systems than we do in artificial systems. And when I look at where nanotechnology is going, I look at those things in which living systems do much better. Things like materials, synthesis of materials, enabling chemical reactions to proceed, energy conversion, a very important problem in society. This is a potentially big impact area. And, of course, chemical sensing. And a good example there is if you try to find truffles underneath the soil in northern Spain or in France, you better use a dog or a pig rather than some sort of artificial sensor if you want to find the truffles. Or if you want to find a buried ordnance or mine. The point I would like to make in my presentation is that high functionality requires high levels of structural complexity. This is certainly true in living systems. It's certainly true in the electronic systems that Dario just talked about. And that it's also important, I like to make the information connection, namely that structural complexity represents information. The information is in the structural complexity. A DNA molecule has information, has the information of my heredity, and it has that information encoded in the way in which the base pairs are arranged, in the structural complexity. Now, of course, a nice example of structural complexity is, in fact, the integrated circuit, and this is something I borrowed off the web from Intel. This is a cross-sectional view of the wiring, and this is a top view. Actually, the integrated circuit today is so complicated that it's not possible in a single micrograph to illustrate this complexity. The only way we can do it is to say, well, these microprocessors now today involve almost 1 billion transistors. Now, there are, outside of the integrated electronics, there are other things, other areas of science and engineering which are coming along at a rapid rate that most of the public and most of this audience may not be aware of. This is an example of what we call a photonic crystal. It has structural complexity. These are built using a lithographic process, which I'll mention in a few minutes. The idea here is to try to utilize with light particles, with photons, with light, functions that of manipulation which may rival in certain circumstances what happens electronically. The ability to manipulate light inside of high structural complexity, man-made structural complexity, is important from the point of view of research. It's an exciting area of research, and the future applications, who knows? We can certainly envision many important applications in telecommunications and in information processing. But the real future remains to be seen. But this is a new paradigm, a new approach, namely of manipulating photons. And this has been available to us. The ideas for this have only been available for something like 10 or 15 years. So since high functionality is dependent upon structural complexity, how do we achieve structural complexity? How do we put three-dimensional structural complexity into assemblies of nanoscale elements? Because that's where the future is, in my opinion, and that's certainly been where the past has been in electronics. High functionality through structural complexity. How do we do it? Well, the planar fabrication technique is what we use today. And that consists, some of you may not have ever seen this before. This is what we mean by lithography in the planar process. In order to achieve some sort of a pattern, a relief structure, or some doping profiles or metallic structures on a surface, the way that's done is the pattern is defined somehow, and the substrate of interest is coated with some sort of radiation-sensitive film. It's then exposed to some pattern of radiation, which then enables a chemical change to take place so that we can achieve some relief structure in what we call a resist. And then that pattern is transferred by an etching process, a growth process, and so forth. Now, actually, this diagram doesn't exhaust all possibilities, but this gives you the general idea of the planar fabrication process. Sometimes we use the term lithography to capture this whole process. As you'll see, it operates on a surface, and it creates two-dimensional complexity. Two-dimensional information. This is how the semiconductor industry works. First, a so-called mask is made, which contains the information. This uses an optical or a laser system. That photomask is then used in a lensing system, similar to a photographic system, or operating a camera in reverse, or what we used to call a photographic enlarger. This mask is then, the pattern is then projected onto a silicon wafer, and then other processes like etching are carried out to produce the chip, which then goes into the consumer product. That's the process. That's the process which has revolutionized our lives. And I attended a talk recently by Horst Sturmer, one of the Nobel Prize winners for the quantum Hall effect, and he said, you know, if we didn't have silicon, we'd use an oxide as a nice material for semiconductors, we'd use other semiconductors. But the one thing that we could not have done without for the information revolution was, in fact, lithography. And that was interesting, coming from Horst. There are many forms of lithography. The one I just showed, the one that's used in the semiconductor industry, is only this one here. These are other forms of information, creating complexity in two dimensions that are subjects, some of which subjects of academic research. Some are in, like some of these things are actually used in certain industries. This is the only one that's used in the semiconductor industry as of today. Some are marked in red, and I call them maskless, and that means that they can create patterns rather than just replicate patterns. The structure, the process that's used in the semiconductor industry, this actually only reproduces the pattern here onto a substrate. So it's a copying process. There are what we call maskless processes are creation processes, creating information, not just transferring information. One of the processes which was of lithography, which has probably the highest resolution and very low cost, extremely low cost, is the process of nanoimprinting, which was invented initially by also one of my students, Steve Chu, who's now at Princeton University. And what you see here is structures at the 5 nanometer, even sub-5 nanometer scale. This is actually a reproduction process from a mask to a substrate. And it's still, the challenge remains then how to create the mask, which is then transferred. This nanoimprint process is actually the same process that's used to make DVDs and CDs. It's the same process, in fact, essentially, that Thomas Edison used over 100 years ago to make records. It's basically a soft material or a quasi-liquid material is pressed with a mask of a relief structure, and then that is made a solid pattern, and then transferred into a substrate. This depicts that process of nano-imprinting. There's really two forms, the thermal form and the photocurable form. Here's the pattern that we want to copy. It's pressed into this material, which is soft, heated up, and then pull away, and then you transfer the pattern into the substrate. And this is in which, instead of a thermal process, you convert this liquid into a solid, a polymer, by means of ultraviolet photons. So these are very simple processes that can be conceptually simple. They can be made more complicated, of course, if you want, but they can be carried out at university laboratories and are very important for research. Now, this is a process of lithography that's been developed at my lab and that Dario worked on in his PhD thesis. And it's a very new approach. It's an entirely new approach, what we call zone plate array lithography. The reason it's new is because it's enabled by some of the products of this semiconductor revolution. The idea is we have an array of lenses. They're actually diffractive lenses, so-called zone plate lenses, that focus incident light into a focal spot. Upstream, we have what's called a spatial light modulator, and these you can think of as little mirrors. They're actually not mirrors, but work something like that, that can turn a beamlet of light on and off. So we can control the on and off of light to these individual beams. Then we move the stage in a controlled and an intelligent manner with the beams, the little spots going on and off, to create a pattern of arbitrary geometry in a so-called dot matrix fashion. Now, this was not possible years ago because the appropriate lasers didn't exist, because the spatial light modulators didn't exist, and because the ability to move a stage with nanometer precision didn't exist. All those things now do exist, and they can be put together in a new process which enables us to write patterns of arbitrary geometry in a dot matrix fashion. Now, because this is a way in which information for structure can be converted from a computer, which then controls the spatial light modulator, onto a substrate, we believe that this is an important enabler for nanotechnology research and ultimately nanotechnology production. So we've spun off, we've demonstrated this in the university and have spun off a small company to manufacture these and to make them available to researchers initially and then ultimately in industry. This is what we call the roadmap for the development of the zone plate array lithography. This just shows minimum feature size as a function of the wavelength of the radiation used. We actually use a 400 nanometer laser, and this shows the results, basically something like 1/3 the wavelength of the radiation. Of course, we can use water immersion. And this shows the important convergence of basic research. One of the students in the laboratory has invented a technique he calls absorption modulated optical lithography, which takes advantage of a certain nonlinearity, which the computer simulations now show that even operating at 400 nanometers, we should be able to produce feature sizes down at the 20 nanometer scale, down where certainly electronics is reaching its apparently limits. And by going to shorter wavelengths, we should be able to get down to the molecular scale. And this may actually then enable us to operate at the molecular level. Now, lithography is nice, but it has some limits. And this shows some actually very low-cost lithography, producing 13 nanometer structures. And at first, it looks pretty impressive. But then if you look a little bit more closely, you'll see that this post here, these are posts in silicon, and this one have different dimensions. This one's very skinny and starting to bend over, and this is vertical and upright. And that's because lithography processes use the arrival of particles, photons or electrons or ions, to create these patterns. And the arrival of those particles has a certain randomness to it. So there's a certain, we use a fancier word in science, it's a stochastic process. And therein lies a problem. So the problems of lithography are the stochastic or the quasi-random arrival of particles, and this leads to rough edges and some limitations on the control of dimensions. And lithography doesn't really allow us to reach the molecular level, which is really the 1 nanometer level. We're at the with the 20 nanometer or 15 or few nanometer level. Also, certain forms of lithography use ionizing radiation, which can produce damage to important biological molecules and other organic molecules. So what are the possible solutions? Well, in my view, we have to build bridges to the molecular domain. And I'll show you some preliminary experiments in that direction. One step is what we call templated self-assembly. To use, Dario showed some of that actually, where you have a lithography process, then you combine that with molecules that like to organize, but don't really do it over a long range. Don't do it with what we call long-range coherence. And the coherence is introduced by the lithography, and then the self-organization takes place on a very small scale of a few 100 nanometers or so, and then we get long-range coherence. And I think there's a great opportunity for invention here. There's things yet to be invented. It's how to directly manipulate the molecules in a way that's production compatible or volume compatible, not by taking a probe and moving around individual atoms, which we've seen a few times. That's interesting, but I don't think that's the way we're going to revolutionize or create a new industries. The way is to somehow develop some inventions that don't necessarily simulate, but achieve the same kind of results as take place in living systems. After all, in living systems, we can reproduce something that's priceless, namely another human being, by a process which involves no skilled labor, if you think of it. Okay. So, some examples of approaching the manipulation of molecules. This is work with a colleague of mine, Francesco Stallacci at MIT. These are DNA segments of DNA. We know how to make those, to which you attach a thiol group, a group containing a sulfur atom which bonds to gold. And you can change the pattern in the DNA as you put these things down. You can then combine in solution the complementary strands of DNA. DNA likes to stick together, like in this fashion. And then with this thiol group attached to another surface, and then you can do what's called dehybridize, to separate the DNA. This is a very familiar process. It's used in so-called PCR.
All that is in a dry or wet environment?
This is done in wet, but this is dry. And then you have a reproduction of the original surface. Now, this shows some experiments that we've done using lithography to produce the 50 nanometer lines and spaces in gold, and with a little ridge, actually even finer features than that, and then attaching DNA in this fashion. And this can be done to this molecule. You can attach other functionalities which produce some fluorescence. This is not a product. This is just playing around, if you will, in the laboratory with lithography and macromolecules. These are some other experiments that Stallacci has done on nanoparticles of, say, gold, that, say, a few nanometers in diameter, attaching molecules, some of which are hydrophobic, some are hydrophilic. And it turns out that they naturally form this striped pattern by so-called energy minimization. Now, Francesco has pointed out a very interesting thing about putting organic molecules on nanoparticles, namely that when that happens, you naturally have a pole at the top and at the bottom. You have always two poles. It's a rule of the spherical geometry. In fact, here's an atomic force microscope of such a pole. This is a depiction. That's the actual micrograph. Now, what that means then is, and he's demonstrated this, is he can pull this particular molecule off the pole and substitute another one at that pole. And then we know how to manipulate the nanoparticles with lithography and, I don't want to go forward, and in that way begin to selectively manipulate a given molecule using the poles of these nanoparticles. Again, what good is it? Nothing that I can think of, but except that it's a learning process. It's a step towards manipulating, working with, creating complex structures with the molecules themselves. Everything I've said so far really had to do with two dimensions, and there's, of course, living systems are three-dimensional, but are they really? If you look at the brain has this highly convoluted structure. If you look at for example, the mitochondria inside of a cell, which is the energy machine, so to speak, it achieves its functionality by being highly convoluted. And with that kind of inspiration, we came up with this idea many years ago about how to use a planar technology that I showed, which patterns things and creates complexity in 2 dimensions and achieve 3 dimensions. And this is what I believe is the way to do it, is working on a membrane. The membrane can be, say, a micron thick or 100 nanometers thick, and we've actually done some of this. And it can be folded. It can be reliably folded. And if it's patterned in advance by electronics or photonic crystals or whatever kind of, or other molecules, whatever we want, we can achieve a 3-dimensional organization by folding the membrane. Signals then can travel on the surface of the membrane. They can travel through the membrane, and they can communicate signals with the outside world by photons or by conduction, by wave function overlap, whatever, to the external world. And what you achieve in this kind of a configuration is what I call maximum interfaces and maximum interconnects. And when you think of it, that's actually what happens in the brain. That's why the brain works so well, is because of, as Uzi pointed out, because of the interconnections. So this is a topological concept for maximizing interfaces and maximizing interconnects and as a result, maximizing functionality. This is, I believe, how 3D systems will be organized in the future. Lithography is important. It puts information into substrates, and ultimately it's information that's important. It's very important for research and for other applications, for small industries, that we develop low-cost, flexible lithographies. It's an essential ingredient. The tools of the semiconductor industry that they use for lithography, they cost $20, $30, $50 million. Totally out of the question as far as using that in research. Totally inflexible for research and out of the price range. But we can achieve the same results by other processes. Not the production throughputs, but throughputs appropriate for research and for small businesses. I didn't say anything about nano accuracy, but this is another important point. It turns out that the Industrial Revolution really took off when metrological techniques were available to enable the production of interchangeable parts. And the same argument is going to be applied to the nanotechnology revolution. In other words, we have to have nano accuracy. And this is an issue which is not receiving the appropriate attention from the funding agencies. 3D is accessible by a membrane folding. And I believe we very much need a technology that circumvents these limits of lithography and bridges the gap to the molecular domain. And we don't know how to do that. All we're doing is scratching the surface, so to speak, with some of the experiments that I mentioned. But I think the holy grail is techniques which are deterministic, highly parallel patterning with molecules is the breakthrough to come. Thank you.